The, hu- the human gastrointestinal tract is made of a diverse community of microbes, collectively known as the gut microbiome, that play a role in maintaining the host's health. A reduction in the diversity of microbial species within the gut microbiome is linked to addictive disorders such as alcoholism. In our lab, previous work discovered that fecal microbiome transplants temporarily reduced alcohol cravings in rats exposed to alcohol. Essentially, these rats consumed fecal samples containing gut microbes, which led to decreased alcohol consumption. We hypothesize that the reduction in alcohol cravings is associated with the restoration of the gut microbiome by FMT and decreased intestinal inflammation. As a note, intestinal inflammation can occur after periods of recurrent alcohol consumption. We expect to see differences in the diversity of intestinal microorganisms in rats exposed to alcohol before and after receiving FMT. In order to apply these findings to alcohol use disorders in humans, we propose that probiotics may be a therapeutic alternative to FMT and support the growth and diversity of the gut microbiome. We also hypothesize that probiotics can aid in reducing intestinal inflammation associated with alcohol consumption.
For this study, we had 3 experimental groups of rats who received either water or alcohol and received FMT from either themselves or from a pool of healthy donors. Specifically, our 3 groups were water self, alcohol self, and water donor. All of the rats went through a 4-day drinking in the dark paradigm that was repeated 3 times over the course of 23 days. The rats were then given a dose of omeprazole for 3 days after the last round of their alcohol consumption to promote the survival of the bacteria from the subsequent FMT. FMT was administered via oral gavage 24 hours following the last dose of omeprazole. Fecal samples were taken 24 hours before FMT and 24 hours after FMT. The samples were then prepared, pooled, and sent for DNA sequencing. To analyze the content of the microbiome, hits from high-quality reads were aligned, assigned to OTUs, identified using NCBI BLAST, and then tabulated. Relative abundance for each sample was determined according to genus, and we used the Cultivis online tool to generate a distance matrix, PCOA plot, and heat map using Pearson correlation subtracted from 1. As predicted, we found that FMT did influence the microbial diversity. Alcohol caused a randomized dysbiosis of the microbiome, which is demonstrated in Figure A, specifically when comparing the pink group to the yellow and orange groups. We also found that the diversity of the samples appeared to fall into clusters of 3, which is consistent with our predictions. Gender did not appear to influence the gut microbiome, but age, stress, or other environmental factors may have played a role. Figure B further supports that FMT had an effect on the microbiome. Moreover, certain bacterial families, such as Lactobacilliaceae, were strongly influenced by FMT. This finding in particular supports the following probiotics methodology.
For this study, we chose 5 bacterial strains that, according to the literature and our FMT data, showed promise as beneficial bacteria for human health. These strains grew in MRS broths and agar plates at 37 degrees Celsius for 24 hours. The B. longum strain required an anaerobic chamber for this growth process. Once these probiotic strains were successfully grown and cultured in plates, we wanted to see if they could survive in a 10% sucrose solution for 60 minutes. The purpose of this was to figure out how to make the probiotics palatable for voluntary consumption by rats exposed to alcohol without significantly increasing their caloric intake. We were also concerned about how long it would take the rats to consume the probiotics, so we first assessed a maximum time frame of the probiotic strain's exposure to sucrose. We diluted each strain 1 to 10 in 10% sucrose and let the plates sit for 60 minutes before diluting in saline and plating the strains on MRS to measure their survival in terms of each strain's concentration post-sucrose. As you can see in the table below, the probiotics survived better than anticipated. The survival rate for the probiotic strains was high and supports the survivability of all probiotic strains and 10% sucrose for 60 minutes. Our next step is to repeat the dilutions in sucrose to accurately determine the concentration of each strain post-sucrose. These results will be important for finalizing the precise dosage of the probiotic strains for the non-invasive voluntary probiotic consumption protocol for rats exposed to alcohol. Moving forward, we want to explore the other factors that might have played a role in microbial shifts within the gastrointestinal tract of rats that received FMT. Proposed factors include age and exposure to environmental stressors. We would also like to take a closer look at the variation that was observed among different individuals as represented by the sequence data. Perhaps there are differences or fluctuations within the gut microbiome that naturally occur. We are interested in determining the expected rate of change within the rat gut microbiome in order to explore possible reasons for individual variation in microbial diversity. To confirm our finding that gender did not appear to influence the microbial diversity of the gut microbiome within the rats that received FMT, we plan to repeat the experiment. As aforementioned, we plan to repeat the probiotic survival assay in 10% sucrose to more accurately calculate the probiotic concentration post-sucrose. Once we determine those concentrations, we can apply those results to a subsequent experiment that assesses the viability of combining the chosen probiotic strains for simultaneous consumption of all strains by rats. This could be influenced by interspecies competition among the probiotic strains. Findings from these future experiments are important to consider for the voluntary consumption protocol, as well as establishing the applicability to humans.